Why is this not? No.
Can you hear me? I can't
Yep.
hear you. You know, you're supposed to have your mask on during lab, right?
I'm supposed to what?
You're supposed to have your mask on during lab. How's everything going?
It's good.
Good. All right. We're waiting on Rose
Okay.
because Kelsey's not going to be here. You're not here, so Rose is the last person outstanding. So I'm going to talk about exam tomorrow, just a teensy weensy bit, and then I'm going to talk about how the lab exam next week is going to be structured. And then after that, it's just going to be them sitting around looking at specimens. So I'll probably end the thing after that. And it's saying my internet connection is unstable, which is unfortunate, but we'll get through it. All right, I don't know where Rose is, so let's just go ahead and get started. So yesterday, I forget who it was, asked me about testing. I guess it was Kelsey asked me about testing procedures for the lecture, and I explained that I'm not going to ask you to have your Zoom cameras on and stuff like that, but I'll be around for questions. And I want to explain a little bit more about my rationale for why I do this way, because it's been something of a conversation in higher education over the last year. Like, how do we do things like give people tests? So last semester in Bio 133, after the first exam, a student came to me and said, a lot of people cheated on the first exam. And I'm like, okay, who? How? Like, give me specifics. The student wouldn't. And so I graded the exam and looked at the scores, and the scores really weren't any different on that exam one in Bio 133 than they have been for the last 10 years in that class. If you recall, Bio 133, the first test is always a little grim because they're getting used to a different way of learning, they're getting used to a different way of being tested, et cetera, et cetera. And over the course of time, grades tend to go up on exams as you get more used to kind of how things work around here. And so when I didn't see any difference in the grades, in the grade distribution, the average grade, et cetera, I was like, well, you know, if people are cheating, they're not doing a very good job of it because they're not, you know, raking in the benefits of not playing by the rules. But because there was a concern by a student, I attempted to address that during exam two. So I made people be on Zoom. I made them have their cameras on, et cetera, et cetera. And then after that, I looked at the scores on exam two and compared those scores to previous years, because we have a long record of exam two in Bio 133. And once again, guess what I found? There was no difference between exam two scores and the scores that we've experienced in Bio 133, you know, over the last decade or so. So when we got to exam three, I was just like, you know, it seems to me when I look at your body language on the camera and stuff like that, that just being on camera while you're taking a test seems to stress you out a little bit. So I just said, enough. You know, if you're cheating, we actually detected some cheating last semester, which is unusual in that class because it was just so blatant. I was grading along and read some answers, and one of them had a really unusual example, and then I read another one that had the same example, which then made me go back and look at the two tests. It's like, okay, one of you gave your test to the other person and you basically copied verbatim. It's like the least creative, laziest form of cheating ever. So we dealt with that. We filed the honor code violation, et cetera, but I don't think necessarily it was a widespread sort of thing. So why stress students out? Why not trust them, et cetera, when if they are doing it, it's difficult to detect. And if they are doing it, it's actually not really gaining them anything. So I just told myself on this front, you just need to chill out and worry about other things. Plenty of other things in the world to worry about at this point. You can go through life thinking the worst of people and nobody is ever going to pull something over on you. Or you can go through life granting people a little grace and a little bit of trust. And occasionally you will look like a fool in their eyes, but it also then doesn't color every interaction that you have with people. And the nice thing about looking like a fool in their eyes is you never actually know that you look like a fool in their eyes, because in order for them to think that you're a fool and for you to know about it, they have to come up to you at some time in the future and say, oh yeah, I cheated on that test in Bio 133 and you didn't catch me. Aren't you a fool? In which case you've just admitted to cheating on an exam. So in one way, I don't gain anything from adopting a stance of distrust. And I potentially lose a lot of things in adopting that stance. Now, you might also be saying, well, you know, you catch plenty of people plagiarizing. I'm like, yeah, well, because plagiarism is something that is rampant. I think plagiarism is way more common than cheating on an exam. But once again, I don't walk into every paper that I read looking for examples of plagiarism in student writing. I'm reading a paper, and then I read a paper and it's written in such a way that it looks like a student is probably plagiarizing, because the most common form of plagiarism that we see in student papers is substitution plagiarism. And substitution plagiarism, basically you take a sentence and then you go through the sentence and you replace words with synonyms. And the problem with doing that, and what makes it so easy and obvious to detect, is that the way scientists use words, it isn't easy to replace synonyms. When you replace synonyms for scientific words with common words that regular people use, the sentence ends up not making very much sense. And then when you start interrogating that sentence that doesn't really make any sense a little bit, you find, oh, you just put in synonyms for these words that were in the original thing, and then all of a sudden you're busted. So I don't go through papers like, oh, I'm going to find plagiarism, I'm going to find plagiarism. I'm reading a paper, and then the paper reaches up and smacks me in the face and says, I just plagiarized. And then my radar goes up a little bit. And then within that paper, I start looking for other evidence, because what I want to do when I file an honor code violation on someone is, in the event that they challenge it, that the evidence that I have for that will survive any challenge. So I don't file honor code violations haphazardly or frivolously. I want there to be a substantive body of evidence within a paper that says, yes, this is worth my time going through filling out the paperwork, and maybe even going through a hearing at the Honors Council to adjudicate this thing. And if I can't find that, if there's like one little thing and it's a little sketchy, I make a note about it saying this is too close to plagiarism for my comfort level, but the rest of your paper looks okay. Pay attention to this in the future, and I'll let it go. So tomorrow, the exam, I'm not going to be watching over your shoulder. I depend upon you to behave like honorable students, and that's where I'm going to leave it, because life is too short to go through life just mistrusting everybody around. Don't abuse the trust. That's all I ask, and I think. The nice thing about an upper level class is I know all of you. So as far as the test next week, Sarah, do you have any questions about that?
No.
Can you hear me okay?
Yeah.
I think I'm a little muffled because of the mask, but. So when you come in next week, arrayed around the lab are going to be the bins. And in the bins, normally I just have one specimen per bin, and I spread them all out, because it may take you a couple of minutes at each bin to deal with that particular specimen. But because I want to keep you spaced out, especially tomorrow, I'm probably going to cluster like 2 or 3 specimens per bin. And there will be a left-hand specimen, a center specimen, and a right-hand specimen, so that at a given bin, you may spend actually quite a bit of time, but then the bins will be further spaced apart. So when you come in tomorrow next week for the exam, you'll see the bins. Everybody will get at a bin. The questions will be numbered. Actually, I should have done this better, more realistically. So it'll be numbered sequentially so that I can grade it. And there's just the form for it I will provide you, and it's just a form with a bunch of numbered spaces with blanks after them, and you just fill in the information for each of the questions. And almost invariably, the first question for any specimen is going to be, what is it? When you give me that what is it, I'm expecting a scientific name, genus, species. Be sure and underline the genus species, because that signifies to me that you know that species names should be either italicized when they're typed out on a word processor, or underlined if you're handwriting them. I may ask you something about the higher level classification. So this is what species it is. What family does it belong to? You know, it's Eurecia lucifiga. Well, what family does that belong to? The Plethodontidae. So that's that. Where would you find Eurecia lucifiga? What's its geographic range? The Ozark Highlands. It's associated with caves and these spring-fed streams in the karst region of Missouri. So the Ozark Highlands. And then where does this organism lay its eggs? Mainly because it's a Plethodontid salamander, and this is something that is kind of characteristic of many of the species in the plethodontids. Plethodontids tend to lay their eggs two places. Plethodontids are known for laying their eggs on land, where they go through direct development. And then there are plenty of plethodontids that lay their eggs in streams. And this happens to be one of those species that is associated with caves. And oftentimes the caves that it is associated are caves that have streams flowing through them or out of them. And they will lay those eggs in the shallow water that is in those streams flowing. So this is one of those species that is going to be laying its eggs usually in slow flowing water. It's not a pond breeder. It's not like the ambystomatids that lay their eggs in ponds, or any of the frogs and toads that we're looking at that lay their eggs in ponds. So, and I'll give you a choice of terms to put in there. And so in that way, it's kind of a multiple choice question. So wherever you start, the first thing I'm going to tell you is circle the number of where you're starting so that you put the answers in the right spot, so that I don't then have to go and reconfigure the key as I go through there. And then you'll just start working your way through. If somebody is at a station that you want to go to, you either stand there and wait, or skip the line. You don't have to go through it linearly, although it will be laid out linearly. But if you do skip over things, make sure that you're putting your answers in the correct place, because you don't want to get graded wrong because you put your answer in the wrong place. You want to get graded wrong when you actually got something wrong. So keep a mind on where you're writing your answers. Make sure that your answers correspond to the number of the question that you're answering. So then you would go to question 5, which is on the next station. What's the name? In this case, what's the order? So let's say that let's say that we have Bufo woodhousei. If that's the name of the species, what order is Bufo woodhousei in? Not class, which would be amphibia, but order, which is below that. We're only dealing with 2 orders in this class because, Say it again, I can't. Yeah, this is order in there. It's the frogs and toads. So that's the order. And since it's a buffonid, what would be a good diagnostic character for this family? So I asked you about the order. I didn't ask you about the family, but the Onyxaris woodhousei is in the family Buffonidae. What is a good diagnostic character for the family Buffonidae? Yeah, the presence of those parotoid glands, or a combination of prominent cranial crest and parotoid glands is even better. But I would certainly take parotoid glands. So for any specimen, expect anywhere from 2 to 4 questions about that specimen. They will deal with, in some cases, anatomy in terms of diagnostic characters. In some cases, they will ask you about higher order classification. In some cases, they will ask you about various aspects of natural history, like reproductive mode, what time of year things tend to breed. Most of the species that are in here are going to be things that breed in the spring. So what I would do is I would just make sure that you know which ones don't breed in the spring. There are plenty of species here that breed throughout the warm season of the year, depending on, for example, when rainfall comes. But most of these things are going to be spring breeding, because that's when temperatures are warm enough that they can be active. That's when rains tend to come predictably in Missouri. And as those rains come, it fills up these temporary bodies of water where these amphibians breed. And you need to breed early enough in the year that your larvae have a chance to develop before that temporary pool of water dries up and goes away, trapping your tadpoles, your larvae in there. So that's how most amphibians in Missouri breed. There are some that don't. So for example, certain of the buffonids, they will breed throughout the year. and largely dependent upon when torrential rainfall occurs. So those are the kinds of things that you need to be prepared for. Now, you may say to yourself, well, how am I going to ever do this if I forget the name of the specimen? Well, two things will happen. So if you can't remember the scientific name, but you can remember the common name, I'll accept up to 4 common names in place of scientific names. That's pretty standard practice in these types of exams. There are plenty of times when I'm looking at a specimen, I'm like, oh crap, what's the scientific name? And I'll remember the common name, but I'll have a brain fart on the scientific name. So you can use up to 4 common names if you want, but most of your names should be scientific names. But even if you may forget the name of the specimen of that particular species, you probably can remember what family it's in. And sometimes knowing what family it's in provides you clues as to what the other questions are. So don't think that just because you don't get the species correct means that you're going to be missing everything about that species on the exam. And one of the things that you will also find is if you can't grasp a name, just circle that question, leave it blank, and just make sure that you come back to it again before you leave. So what I'll do is I'll encourage you to go through things in order, but then there are always going to be some things that you want to go back and look at a second time. While you're doing other things, your brain will continue to think about that specimen even without you thinking about it, and it may pop into your head what that specimen is. At that point, stop whatever you're doing and write that down. You can write it in the margin, you can write it in the place where you think it belongs, but then go back, jot it down somewhere so that if it then escapes your brain again, you have a record of it. And then go back and check to make sure, oh yeah, that thing that popped into my brain, that is really actually that specimen. The other thing is if you forget the name of one thing, let's say you're working on the Bufonids and you get to one Bufonid and it's like, oh my gosh, which one of these Bufonids is it? And then 5 specimens later, you see another Bufonid and that Bufonid resonates with you more than the first Bufonid did in terms of what it is. And then it's clear to you, oh, this specimen is clearly showing the characteristics of this species. Once again, in the margins, jot down a couple of notes. Okay, this is what made this species this, so that when I go back to look at that other species of Bufonid that I couldn't get, I have some clarity as to what the other Bufonids look like, which then helps you go back and get that name. So it's not you get one shot at a specimen and then you're over with. You can go back and revisit any of the specimens that you want again. And oftentimes the process of seeing other specimens jogs things in your memory about specimens that you were maybe a little sketchier about. So think of the exam as a process rather than just a kind of a one-shot deal. At the end of the exam, I will collect all of your papers, and then I'll have the key to the exam, and we'll actually go through the key to make sure that the answers that I have to the key are the answers that you provided as well. So even though it's not graded at that point, you should have a pretty good idea of how you did by the time you exit the room. And then once the exam is over, we'll put all those specimens back, and I will have already put the reptiles out, and then we'll spend the rest of the time beginning to work on reptiles. Questions about that?
How many specimens do you think will be on the test?
So it's usually got about 50 questions. So 50 divided by 2 to 4.
Okay.
So, and with a limited number of species, chances are you're going to probably see every specimen, every species that we have in lab right now. That won't necessarily be the case with the reptiles, but it's probably going to be the case with the amphibians. Kara asked how many species should we expect, how many specimens should we expect to see? And somewhere between 20 and 25 probably. Yeah. Yeah. So what I'll probably do, if I have 3 specimens in a bin, here's the bin. If I have specimen 1, specimen 2, and specimen 3 here, I'll put a card here, I'll put a card here, and I'll put a card here. This one goes to this specimen, this one goes to that specimen, this one goes to that specimen. So it's clear what's going on. The other thing is the specimens are going to be out for a while, so they're all going to be covered with paper towels. They're moistened with ethanol. I also have ethanol containers around. If you see that your paper towel is beginning to dry out some, just grab a bottle of ethanol and re-moisten things. I'll probably be walking around the room moistening things as we go as well. So that's the other thing. So to do this effectively, it's imperative that you don't move cards and don't move specimens. So once again, kind of like the way you're studying, if you're looking at the toads, for example, be careful in how you handle the toads so that you get the toads back in the right bottle so that somebody doesn't come along after you and pull a toad out of the Bufo americanus or the Anaxyrus americanus section that's actually Anaxyrus woodhousei. So keep them in their order so that when other people come to that bin, they are looking at the same species in the same order that you looked at them in. But during the test, you're totally free to pick up the specimens, look at them, turn them over. For some things, you have to turn them over. For example, toads, you need to see what the spotting is like on the underside, so you can handle the specimens the same way that you would handle the specimens here in lab. So you might want to have one hand gloved and one hand ungloved. One hand gloved for handling specimens, and the other hand ungloved for writing, if you want. Other questions? Rose, you came in late. I'm on with Tiara, so I'll post this to the YouTube so you can see what we talked about before. Basically talking about testing procedures for next week. All right. Other questions? Tiara?
No, not about the test.
Okay. Do you want to stay on the line and talk about the dichotomous key for a minute, or do you want to do that some other time?
That's fine. I only have a couple questions.
Okay. All right. You guys get to work. Kiara has some questions about the dichotomous key. And, I brought some blank note cards. So the questions will be on these little note cards. So if you want to make up quizzes for one another in the same way that they'll be kind of done on the test, there are plenty of note cards hanging around for you to do that. Okay, what's up, Kiara?
I had a question about the tree frogs, the gray tree frog and the Cope's tree frog.
Yes.
Since there's no physical characteristic to distinguish them, do we have to distinguish them in the dichotomous key?
No. And so let me talk to the rest of the class about this also. So when you see a gray tree frog on the test, you can either write it out as Dryophytes chrysosceles, or versicolor, or if you want to be a fancy pants, super complete, super knowledgeable herpetologist, you can write Dryophytes chrysosceles / versicolor to indicate to me that you have complete knowledge of what that specimen is. In the same way, because there is no morphological way for a person picking up a specimen in the field to distinguish those two frog species from one another, because you're not carrying around your blood sampling kit and your compound microscope. You don't have to key those out within the key as separate species. You can just key them out to gray tree frog, Hylocrisophilus / versicolor. Does that answer your question, Caroline?
Yes.
Okay.
And
what
then
else you got?
I just had one more question when I was
Mhm.
looking at the chorus frog and the Cajun chorus frog. It said that they're basically indistinguishable, but it was just by call. Like the call has a slightly longer duration, and then it was
Which,
a longer,
which,
a looser pulse rate, I think is what it said, between upless
which
chorus frog and
upland course?
the Cajun chorus frog. I just didn't know if I was missing some kind of physical characteristic between them.
Can I borrow your field guide for a minute? Yeah, so upland versus Cajun.
Yeah.
Okay, give me a sec.
Okay.
This is one of those new divisions that I don't think about a lot. Yeah, so in this case, you would have to put something in there about the call.
Okay.
Yeah, because and you're going to see that this is the same case with the fence lizards. So when we get to the lizards, when we get to Scalopris undulatus and the other members of the fence lizard group, they have been divided up on the basis of molecular markers, but they're almost virtually indistinguishable from one another morphologically. And so it even tells you in the field guide the easiest way to tell them apart is by the geographic range, where you actually collected it. In which case, if geography is a distinguishing characteristic, then that's totally appropriate to put in a dichotomous key. Yep.
Okay, perfect.
So where's the Cajun chorus frog? I believe it's down south, right?
Yeah, it says southeast Missouri to Oklahoma, south to the Gulf.
Yeah, let me get back to that section again.
Oh, it does say best distinguished using geography and molecular markers.
Yeah, because even the call is not that different. Okay, so in Missouri, it's really limited to down there in that Boot Heel region. Well, so the upland chorus frog is more boot heely than the Cajun, but they're both down in that area. So that's tricky. Yeah, tricky.
Okay, but those were the only two questions I had.
So one of the things that you could also do on that particular couplet, separating those two species, is actually just get a copy of a range map and put that in there if geographic range is the thing that you're going to need to be doing. Yep.
Okay.
And you're going to find yourself doing the same thing with the fence lizards.
Okay.
I was in New Mexico a couple of years ago, up around Santa Fe, and it's a part of the state where you have the potential to collect 3 possible different species of fence lizard, and it's like, geography. I'm like, oh yeah, but all 3 of them, they had, like, all of their ranges meet up right there in the Santa Fe region. So it's like, it was a crapshoot because they are virtually indistinguishable. So yeah, okay, good.
Perfect. Thank you so much.
All right, see ya.
Bye.